morning, everyone. My name is Dr. Jason Chin. I'm from the National Cancer Society in Malaysia. How do you do? I hope you guys are doing okay on this fine Saturday afternoon. So today we've got an exciting lineup for you. Today is a little bit interesting. It's different from what our usual talks will be. Today we are actually going to be having a talk by a wonderful professor from Genomics, MGenomics. Right? It's a very interesting company that will tell us about demystifying cancer. Right? They will tell us everything to do regarding cancer and everything to do regarding genomic and molecular studies. But before our lovely Professor Manikam Ravichandran gives his talk, let me introduce him just for a little bit. All right? So, Prof. Manikam Ravichandran is the Chief Scientific Officer in MyGenome, Sdn Bhd. He is the Dean of Faculty of Applied Sciences, Ames University. He obtained his MSc Medical Microbiology from Christian Medical College in Vellore and gained his PhD in Biotechnical degree from Anna University, Chennai, India. Currently, his research interests are genetically engineered cholera vaccine and formulation, molecular diagnostics, and phage therapy. He has 77 publications in international journals with a cumulative impact factor of 152 of 950 citations. He has supervised 40 postgraduate students, filed 8 patents, and commercialized 3 products on diagnostics. He has been awarded 44 national and international awards for academic and research excellence, including Ideas, Inventions, New Products, IENA, Nuremberg, Germany, International Exhibition of Inventions, New Technologies and Products, Geneva, and Anugerah Inovasi Negara, Malaysia. He was an associate member of the committee member on the Top Research Scientist Malaysia, TRSM, of the Academy of Sciences Malaysia, ASM. He is a former Vice Chancellor of Ames University as well. So without further ado, please give a warm welcome to our very young-looking Prof. Manikam Ravichandran.
Okay, thank you. Thank you, Dr. Jason, for a kind introduction. Good day to all the doctors and all the participants. Let me share my slides. Excellent. Good day to everyone. And today is an interesting talk on cracking the cancer code, demystifying the fundamentals of genomics in the field of oncology. Actually, we are very lucky to be in this current era, genomics era, where a lot of revolutionary cancer treatments are ongoing. We are actually in the threshold of the cancer treatment with a lot of emerging tests on genomics. I am not here to present about the clinical side. I'm just going to demystify on the fundamentals of gene and genomics, how these tests are done, how we can choose for the diagnosis. That's my aspect. And I'm basically a clinical microbiologist turned into a biotechnologist and a genetic engineer. So let me start with this. This is the overall content of my slide. So I will briefly talk about what is cancer, what is cancer cell, and what is the genome and how the genes are organized, and a little bit about the mutations, some basic thing we need to learn. And most important thing about how to differentiate the germline mutation and somatic mutation. So this is very important point. I want everyone to keep attention on it, and different type of cancer genes. I'm not going to cover all the cancer genes, just few representative genes. And most focus is on type of genomic assays like whole exome sequencing, whole genome sequencing, targeted gene sequencing, circulating tumor DNA, and epigenetics. So cancer is a disease in which some of the body cells grows uncontrollably and spread to other parts of the body. And cancer can start almost anywhere in the human body, which is made up of trillions of cells. So that is the different. The cancer cell grows uncontrollably. That's all about what you want to, how you want to detect it and control it. That's all about it. It's a non-communicable disease, as we know. It is actually a genetic disease. There is a change in the genome, makes the cells to be not eat. So the genome of cancer cells keep changing. It is different from the normal cell, and they are unstable. And cancer cell mutate from time to time. And sometimes if you take a cancer tumor, the mutation may vary from cell to cell, so location to location. So it is, in a nutshell, cancer is a genetic disease. Okay, we need to understand what is a cell. A cell has a nucleus. Inside the nucleus, you have the genome. And genomics basically is the study of genetic sequence information of an organism. And the genome present in the cancer cell is called cancer genome. There are many approaches for cancer treatment, like currently we have all this chemotherapy, radiotherapy, and surgery. But with the results of cancer genome and the mutation found in the cancer genome, we have the targeted drug therapies available. So basically, each mutation is matched with the targeted therapy. So that's how the whole thing is done. And I will not be focusing on the mutation and the drugs, but we'll be focusing on the methods. So any living organism has a cell in them. And irrespective, we are not focusing on all these things, but we are focusing on human. So inside the cell, we have the genomic DNA. In human body, we have 30 trillion cells. We have a lot of tissues and various organs, and we have cells. So each cell is a building block of our body. So from each tissue, you have cells. Inside the cell, you have nucleus. Inside the nucleus, you have chromosome, which has a P arm and Q arm and centromere. Inside the chromosome, you have this double-stranded DNA, which is made up of up to 3 billion bases. 3 billion bases, basically made up of 4 nucleotides: A, T, C, and G, adenine, thymine, cytosine, and guanine. And these all are present, spread across this 23 pair of chromosome, and where the genes are located. So just a little bit about how genes are coded. They are coded by codons. And the 3 letters I mentioned to you, A, T, G, and C, they are in the combination of different combination codes for different amino acids. I'm sure you all already learned about it. I want to demystify here. ATG, for example, this is a small gene. It starts with ATG, CAA, and then ends with TGA. TGA is the stop codon, ATG is the start codon, methionine, ATG codes for methionine, CAA codes for glycine and lysine, and so on. So what happens in the cell is if there's any change in one of the base here, it codes for different amino acids. Basically, protein is made up of chains of amino acids. So when there is a change, the protein structure changes, the function changes, and sometimes it can be no change, but most of the time it can lead to some deleterious effect. So this is just an overview how the chromosome looks like. We all know chromosome. Chromosome has packed DNA with histone molecules, and several genes are located in these 30 billion bases, which is there in our genome. And you have to remember, not all genes are expressed all the time. Imagine we have 22,000 genes. If all genes expressed all the time in all the cells, will be like a bundle of muscles, a bundle of mass of tissue. So each gene has its own role. They are positionally expressed. That's very important. That's the beauty of our human body. Though we have all the 22,000 genes in each and every cells of the tissue and organs, but the genes are, for example, insulin only expressed in the beta islets of pancreas, melanin only expressed on the skin epithelial cells, skin cells. So genes are expressed based on only need. So, and every cell growth is controlled. When the growth control is not maintained, then it becomes the cancer. I want to bring everyone to attention of this particular simple central dogma. where this is the whole process. Imagine this is the whole chromosome, the whole genome, let's say 30 billion bases in our body, and which is there in the chromosome. And the genes are expressed only based on demand and need in the body. Okay, so this is the genomic DNA, which has a promoter. These are the controlling elements for the gene to express or not to express. Intron, or the intervening sequence, it has no role in it, but it has some indirect function. And exons are the real genes in our genome. So that is actually transcribed into an mRNA, and the mRNA is produced into protein. So when a cell needs insulin, so it gives a signal, the promoter works, and then produces this mRNA and makes the protein. This is how the whole process works. So it's called central dogma, from the DNA to RNA and RNA to protein. So what we do we might come across many things in genome sequencing. People call exome sequencing. Exome means basically sequencing all the exons in the genome. Human genome has 30 billion bases, of which only 1 to 2% of the whole genome is exons. Around 22,000 genes have exons, and this is what the whole exome sequencing done. And the one uniqueness of human and bacteria, when you compare to bacteria doesn't have introns. They all have a direct sequences, but humans, eukaryotes, the higher organism has introns. And we would have come across whole genome sequencing. Whole genome sequencing, sequencing the whole 30 billion bases. That is the whole genome sequencing. Across that will sequence the 23 pair of all this 100% of the genome. Exome only sequence around 2% of the genome, and whole genome sequencing 100% of the genome is sequenced. And people are some panel of genes we sequence. Okay, targeted sequence is called targeted sequencing. I think doctors would have come across some gene list, or we sequence these genes specific to the cancer. They either sequence the mRNA or only the specific region of the gene. That is called targeted sequencing panel. And epigenetics is basically there is no change in the genetic material, but there is a methylation in cytosine or a few bases in the DNA. So the methylation sometimes can prevent the gene to express, sometimes can overexpress the gene that can lead to cancer. So these are the generally 4 different methodologies. There are so many other methods. I'm not covering the immunohistopathology and immunohistochemistry, ELISA, PCR, microarray, so many other methods are there, but these are the genomic technology which I'll be covering in this lecture. Mutation is actually defined as a heritable changes in the nucleotide of an organism. It is estimated that the mutation takes place in human cells 1 out of every 10 billion cell division, because our cell has to divide, we have to grow, and we have to rejuvenate there is a wear and tear, and we need to do it. But the cells our human cells are very accurate. We have a proofreading system. We have our DNA polymerase has a proofreading capability because we have double-stranded DNA. If there is an error happens in other strand, the other strand knows there is something there, that enzyme recognizes and then corrects the base. So always we have the proofreading DNA polymerase in human cells, and that corrects the error during the replication of the 30 billion bases. Imagine we ask you to write 30 billion letters in one page. In every page, sometimes we tend to do mistakes, but we have erasers to correct. Our human cells has DNA polymerase, which has the capability to correct the error. Only when that fails, then we create the mutation get accumulated that leads to other problems. What causes mutation, like tobacco use, ultraviolet radiation, viruses, and aging? These are some of the causes of mutations. And this is just, don't worry about too many bases here. There are types of mutations. For example, the codon here is CCA, which codes for proline amino acids. When that is changed to C to T, it becomes CTA codon, which codes for leucine. So let's say it's a protein supposed to be in this place, we have proline amino acid, it will become a leucine amino acid. And the transversion is basically, the T is changed to A, So instead of phenylalanine, it changed to tyrosine. Sometimes there's mutations, a silent mutation, because there's a change there, but both codes for the same amino acids. So there is no, it's called silent mutation. So sometimes you can get a deletion, one part of the gene can be deleted. And if there is one base insertion, the whole amino acid downstream will be changed. Let's say you have a protein, the first N-terminal region is okay. If there's one base deleted or added, inserted there, the following one will be totally different. It will be entirely different protein. And so this is types of mutation which I described before. So this is actually missense mutation. There's a change in the single DNA base that results in amino acid sequence change. And frame shift mutation, either by insertion or deletion, it can lead to changes. Nonsense mutation, which what happens is instead of amino acid, it codes for a stop codon. It stops the chain of the protein. So protein is supposed to be 100 amino acids. Suddenly it stops at 10. So it becomes a small fragment of protein. It doesn't have function. It's supposed to do certain important activity. The whole cell might get damaged. And in humans, not only the micro change at the bases, we also have a structural change in the arrangement of the gene, such as chromosomal rearrangement. During cell division, maybe the one P arm can bind to the another chromosome. There can be fluctuation in some of the whole arm of the chromosome can fusion and so on. Sometimes gene get fused. What happened is one part of the gene will be there, the other part of the gene get fused. Whole genome sequencing can identify these kind of fusions and kind of changes. And there are some terms we tend to use. It's called SNPs. It's called single nucleotide polymorphism. Any changes in the genome is called polymorphism, and it can be due to deletion or substitution or any changes. And for example, this is one of the SNP, a single nucleotide polymorphism at a particular location. So if you can see here, G allele is in one of the two copies, in another allele, which in Karen has A allele. So this single nucleotide polymorphism varies, and different individuals have different genetic variations. This one we call it as a genetic markers. And these genetic changes can lead to one of the things we are talking about it today is the cancer, a risk of a disease. Sometimes it can associated with the wellness and fitness condition. Sometimes it can associate with drug response, which is called pharmacogenomics. And it can also lead to adverse drug reaction and so on. So when we do the sequencing for cancer, actually you have to remember the cancer is present in the human body. So we have two type of things. One is the cancer cell, and another one is the normal cell. So any changes we see in the normal cell, normal human body is actually the cells are present in every part of the body, and they are called germline mutation. Any changes in the normal cells in the body is called germline mutation. They are inherited mutations. It comes from the father and mother, and it's passed from parent to the child. And inherited gene exists in every cells in the body, and it is, you can, based on the mutation, you can plan the targeted drug therapy and so on. Normally, how do we do this test? We either take the buccal cells using a swab, saliva, and then do a genome sequencing, or through the blood sample from the patient. So that's a germline mutation. Germline, if you take the blood sample or from buccal swab, the sequencing results are basically for the germline. Somatics are basically on the cancer cells. As I told you, cancer is a genetic disorder. Even if you take a tumor, the changes are dynamic. It keeps changing. It starts with one main mutation and then keep on changing. So these are acquired mutation. It will stay only in that particular part of the body. It will not be inherited to the next generation. The somatic mutation will not be inherited to the next generation. It's acquired over a lifetime of a person. It is not passed to the child and exists only in the tumor itself. And it's also the mutation. Based on the mutation, you can check the FDA approved drug, which mutation match with the drug. I think you can refer to it and then treat it. So the somatic, in nutshell, somatic mutations are the mutations seen in the cancer cell. Germline mutations are the mutation which is inherited by the person which is present in the whole body. There are three types of cancer gene. There are so many types, and I'm just telling you generically why there are the three types of cancer genes that control the cell growth and cause the cancer to develop. Normally, a cell has cell cycle. It has to go through different phases of this life cycle, and it knows where to stop and where to grow. So, and the genes also positionally expressed when it is needed, genes are expressed. These oncogenes is out of control, basically. So oncogenes are the mutated gene that cause cell to grow out of control, can lead to cancer. Proto-oncogenes are normal genes. They normally control the cell growth. If they are mutated, the cell growth out of control and can lead to cancer. Tumor suppressor genes are normal genes that slow cell growth and division and repair the mistake in the cells when to die. Cell has its own mechanism called apoptosis, and it's called programmed cell death. So they help to protect against cancer. When the genes are mutated, they grow out of control, that can lead to cancer. The third type of gene, I tell you about the proofreading before. The DNA repair gene that fix mistake in other gene that happen when DNA is copied, DNA repair genes are mutated. It can't fix the mistakes in the oncogene, and that can lead to the cancer. These are three types of gene: oncogene, tumor suppressor gene, and DNA repair gene. There are many cancer genes there. Some examples are TP53 gene mutation, DNA mismatch repair genes, and adenomatous polyposis coli genes. There are several genes, and basically, as I explained before, either they are oncogenes or tumor suppressor gene or DNA repair genes. How genetic mutation lead to cancer? Actually, body has its own mechanism to repair the cancer cells, and we also have an immune system, very strong immune system. We have in our body, we have something called natural killer cells. Whenever there is a mistake happen in any cells, and our immune system clears them away. Number one, we have our own proofreading mechanism to repair the mutation. Number two, we have our own immune cells to monitor any abnormal cells and kill them on the spot. But what happens in cancer cells, it has a first mutation and it has second mutation, and slowly the mutation get accumulated, and the cell tumor, it start growing. What happens is that when the mutation happens, the cell grows, uncontrolled cell growth lead to the tumor formation, and the tumor invade the surrounding tissues, and the cancer cell tend to travel through the blood vessels. They metastasize to the other organs and then start growing. Secondary tumors are formed in the body. So let's take an example of a colorectal tumorigenesis. So in this process, you can see here, the mutation happens gradually. It's an accumulation of mutation. So it's processed through a series of genetic alterations. These alterations include RAS gene, tumor suppressor gene, particularly on chromosome 5q, 17p, and 18q. Each stage involves mutation on one particular gene called APC gene. So also the alteration in DNA methylation also follows, then KRAS mutation, and so on. So it's basically accumulation of mutation that leads the normal cells into cancer, and then finally lead to carcinoma and metastasis. There are several genes linked to the hereditary cancer, like breast cancer, colorectal, endometrial, ovarian, gastric, melanoma, and pancreatic and prostate cancer. There are several genes involved. For example, you have breast cancer like BRCA, and all the other genes are involved in this mutation. So there are certain genes are very aggressive, associated to the cancer, and certain genes are in the early stage. So we have to use accordingly. Is cancer is hereditary? I think I told you before. Genetic change happen, the cancer can pass down. It can be inherited only if it is present in the egg or sperm. When there is changes in the germline, it can be inherited. For example, if a parent has a BRCA1 and BRCA2 gene to the child has a higher risk of developing breast or other several cancers. So it's if you do a germline genome testing, if you find BRCA1 or BRCA2, so you can take the chances are high. It's not necessarily you have a gene that it causes the cancer, but you can take precautions. You have highly prone to cancer, and you can take precautions. Inherited cancer-related genetic changes does not mean that you will definitely get cancer. It means that you have a higher risk of cancer, because in our body, not only the genome, we have epigenetics, we have the environment, the food habit, and all the other things that really controls on top of the genes. So there are many types of genomic tests. So these kind of mutations are happening in various genes. We are not focusing on the genes. So how to detect it? So you might come across a lot of words. I just want to demystify those words for you. Whole exome sequencing, whole genome sequencing, targeted gene sequencing, circulating tumor DNA, and epigenetics. So the whole exome sequencing is a genomic test that only focus on the protein coding regions. So in which I told you, we have 30 billion bases, only 1 to 2% codes for the protein which we need, or the functional elements we need in our body. The rest of the things are repetitive sequences, introns, and intervening sequences, and a lot of other things we have. When we evolved from a single cell into human, we had so many genes which we have genes nearly, you can see that we share around 50 to 60% of the gene with tree, and 80% of the genes, similar genes, are present in cow, and so on. So we have so many genes which we are inherited, and some has function, some do not have function. And whole exome sequencing has its own disadvantage. It has only the limited coverage. It does not cover the non-coding region of the DNA. It is known that some of the non-coding region of the DNA, like intron, does have some function. So it covers only around 20,000 genes what we have, and the cost and the use we have to balance depends on the patient's need. So this is the central dogma of molecular biology, which I explained before. This is a whole genome I represented here. Only 2%, 1 to 2% of genome codes for the gene. So in the case of exome, whole exome sequencing, we cover only the exons that code for the protein and the genome. So basically, there are several methods to capture the exon part using the mRNA, or different methods available. So they code only capture these exon sequences around what this is present 1 to 2% genome, and that is sequenced. Then they will tabulate and tell you what gene has mutation and so on. That's number one. Number two is the second method is whole genome sequencing. Whole genome sequencing is it sequences everything, including the exon, intron, repetitive sequence, everything. The 30 billion bases will be sequenced, and very important. So whenever somebody offer you the whole genome sequencing, ask them for a coverage, how much times it covers. If you get 100 times coverage, that's the best good quality sequence. Sometimes they give you for 10 times coverage, means the coverage means you have to, when we do the sequencing, we need to have many contigs. How many times repeatedly sequence, the error will be less, accuracy will be good. So this provide the whole genome sequencing is one of the best genomic tests, but it's very expensive. It gives you the entire genetic makeup of these 3 billion letters, and I have my genome sequencing book, which is like 400 pages. It tells you everything you're allergic, what you're allergic to, and it tells you the pathogenic genes and cancer-related genes. You have your risk for cardiovascular diseases. Everything is like a book of life. Once you do it, you can keep it with you for your life. Your genome is not going to change, except that cancer, if at all developed, but the genome will be same. So all these thing, whole genome sequencing, basically they sequence. You see the repetition here because of the coverage. It covers 100 times or whatever, so you get accurate sequence. Coming back to my central dogma, so whole genome sequence the entire genome, including all the intron, exon, and the whole part of the genome. So all the 3 billion bases in the cell will be sequenced. It covers all the 100% of the genome, so you get a very good output of the genome sequence. How do they do it? Either they take a buccal swab or a blood sample, they fragment it, create a library, and then do the sequence. There are many methods, Sanger sequencing, and you have Pacific genome sequencing and NGI, and so many different platforms. I don't want to confuse everyone. So what we need is a blood sample or a buccal swab, and send to the company. They will sequence for you. Only thing you can ask them, does it has a better coverage, better accuracy, and so on. Comparing the WES with WGS, the exome sequencing with the genome sequencing. Exome sequencing only covers the 2% of the human gene, and whole genome sequencing is like you read every page in your book, life book, every page, and look for the error, and then finally they give you a VCF file. You know, all the errors will be put in a different file. You can check everything happening in your genome, you know. So there are many studies they have done, and they say the whole exome sequence diagnose only around 30% of the patients. And they say WGS exploring the non-coding DNA diagnosing more than 70. Let's say you have a challenging case, so the whole genome sequencing will give you some answer. If anything, that let's say that we hit the wall, we don't know how to find the problem in the patient, whole genome sequencing might give you some answer what's happening in the genome. So this is just a pictorial view of whole genome sequencing. Exome is just only part of it. Gene panel will be a sub part of it. And whole genome sequencing, as I mentioned in the central dogma, you know each gene have many exons. Some gene have hundreds of exons, and they have intervening sequences and so on. For example, this is gene which has 5 exons. So when it forms the mRNA, it has to form 1, 2, 3, 4, 5 together. What happens is it splices 1, 2, 4, 5, 3 is missing, 1, 2, 3, 5, 4 is missing. It forms an entirely different protein. It can lead to a certain disorder, either organizational complexity or organ development or disease or cancer. So whole genome sequencing can identify the changes because it puts the sequence in order. So you know when the blocks are changed, you can identify them. In whole exome sequencing, the blocks are individually sequenced, so you won't know whether these kind of things are happening or not. So the key purpose of WGS predict the changes in hereditary cancer in a person's lifetime, reveal changes, mutation based on the genome data, and the limitation of liquid biopsy. The test is specifically designed for heritable germline mutation and is not appropriate for detecting the somatic mutation. So WGS normally done on blood samples, so we won't do it on a cancer sample. So that's one of the limitation. So the third test, what we could do is targeted sequencing. Let's say you have a pancreatic cancer patients or colon cancer patients. You can sequence for the specific gene. There are simple tests available such as polymerase chain reaction, PCR, or microarray. They are cost effective and rapid. Immediately you can get it. So it's this is a very focused. Sometimes you have a cancer panel, 100 gene, 600 gene panels available. So you can sequence for the specific genes, and I can look for it. And then based on that, you can go for the targeted therapy. Again, This method is only either look for the mRNA from the gene is expressed, or the particular exons are looked into the genome. So this is actually the targeted panel-based sequencing method. So they use a technology called PCR. What PCR is like a photocopy machine. It copies, it amplifies the specific region of the gene billion times. So we can easy to see, and you can sequence and check the errors in the gene. Microarray is one of the methods where we also can detect the mutation in the gene for a particular point with the probes are fixed onto the panel, and you can detect them. This is some of the examples of panel tests. This is one of the things which we are offering at MyGenome, which will look for the 605 genes in one of the panel, which we recently acquired this panel, and look for all these different type of cancer and diseases. And also there is a cancer screen panel, look for around 26 different type of cancer. It's specific, only the specific RNA expression is looked at this panel. And the reports will tell you whether they are high risk, or pathogenic, or non-pathogenic, or upregulated or downregulated. So this kind of a very kind of a pictorial report, it helps the physician to understand the report. There is a fourth type of thing which is also coming up. It's called ctDNA, circulating tumor DNA, or circulating tumor cells. So they can be detected in the bloodstream because when the cancer grows, they also shed some cells. Sometimes shed the DNA. It will be circulating in the bloodstream. For example, you have some of the cancers very difficult to get blood tissue samples like brain and so on. So this DNA is normally released into the bloodstream that can be detected. So this tumor also develop dead cells that that break down and release the DNA into the bloodstream. They are around 200 base pair in size. Diagnosing certain tumor, as I mentioned before, like tumor in the brain or any difficult region we can access. So you can look for the ctDNA. So ctDNA is basically everything. It can be a part of the genome, or it can be an RNA, or it can it's basically from a cell. It can be a normal cell too. The ctDNAs are circulating in the blood, so detect it. There's one technique I came across. There are so many techniques there. I'm not propagating this. So there is a technique which MyGenome we are planning to acquire this technology. It's isolation of size of tumor cells. I said this is a technology, very, very interesting method. So what they do is they take the blood, maybe around 5 ml of blood, and they filter these cells which are circulating. They are big in size than the normal cells. So the cells are filtered onto this membrane. So once these floating cells are filtered, and you can look for normal histopathology, immunofluorescence, molecular analysis, and so it's a very simple method which we can use to detect floating cancer cells in 14 minutes. So the technology is from France. So that is about circulating tumor DNA. The last technique, which is also very relevant, we know mutation is change in the basis that causes the cancer, but there is also something else which is no change in the base, no change in the DNA basis, ATGC, all same, but there is a change in the methylation. So some changes in the methylation of the DNA can cause cancer. So there is a group of heritable modification without altering the DNA itself. This is called epigenetics. This area is called epigenetics. So it is basically happens due to the methylation in either in the histone region or in some other chromosome region. So this is how the gene has to work. Let's say this is a gene which has a promoter. It has to produce this exon into transcript and so on. What happens is when these methyl groups are added in the cancer cells, so the gene expression either stopped, or sometimes the methylation is removed. It will start functioning fast. So this either over methylation or under methylation will control the expression of the gene. This is called epigenetics. Epigenetics, there are two major types of epigenetics. So one is the direct modification of the DNA strand by addition of the methyl chemical group, particularly in the cysteine, ACTG, the C, the methyl group is added. It can be sequenced by the sulfide sequencing. Chemical modification of the histone protein, that the DNA strands are wrapped around. So the histone, which is wrapped around, also that has some modification. These two can either silence the gene expression or enhance the gene expression, depends on the tumor. Okay. So this is one of the method. The technology to do the methylation is slightly different. Before sequencing, they do some chemical modification with sulfide and then do the sequencing. Then you know where are the places where the sequencing has happened. One of the example of epigenetics gene is that's a gene called SND1 gene. When there is a methylated methylation happen, it promote angiogenesis in human hepatocellular carcinoma, and also this particular gene has a role in cancer. So epigenetics also plays a major role in some of the diseases and cancer, metabolic syndrome, diabetes, heart disease, and rare diseases, and so on. So which sequence to use? That's always a confusion. What kind of whether to go for whole genome sequencing or not. That's I came across few papers that they say the whole genome sequencing. Of course, it covers the whole 30 billion bases. So it can even identify the thing which is not been discovered before. So there are papers which says that whole genome sequencing of colorectal cancer reveal prior treatment effect and specific metastatic features. And WGS, it's actually it can clearly identify the cancer, and it's the most efficient way to give the comprehensive picture of the genomic variation in tumor. And I'll just give you one case report, which was done in MyGenome. We identify a driver mutation in one of the cancer patients. We voluntarily came for whole genome sequencing. So before I start, I have to tell what is driver mutation. Driver mutation is changes in the DNA sequence of the gene that causes a cell to become a cancer cell, because this is the first prime mutation that makes the cell to go into the cancer mode. So then it goes and spread across the body. So we had a case of stomach cancer, stage 3. And the patient had a mutation from a tissue biopsy, that's a somatic mutation and in the JAK2 genes. And then the patient gave this blood sample for whole genome sequencing. In the whole genome sequencing, we found a few SNPs at the MTRR gene. So the MTRR gene, we found that MTRR gene polymorphism has been strongly associated with the gastric cancer, which is it's significantly associated with the poor overall survival and hormonal role in this particular gastric cancer. And also there's another gene called SLC19A1 gene also we identified. It is a major folate transporter. This is very important for the cell growth, particularly. There was a SNP in that, and when you do the literature search, you can also identify the what genotype. By sequencing, we know what genotype it is. Based on genotype, we can also look for the recommended drugs and literature, and we recommend to the patient. So in nutshell, so we have whole exome sequencing, which covers only 1 to 2% of the whole genome, and whole genome sequencing, which covers the whole genomes. You have the whole book of life with you. And targeted sequence, only just use a specific panel, look for the genes. And epigenetics methylation is only a method to identify if there is any methylation as a role in cancer or not. In summary, we have covered only five. I have not covered all the other methods. So only whole exome sequencing, whole genome sequencing, and targeted gene sequencing, circulating tumor DNA, and epigenetics. Actually, this generation, what we are in the threshold of the revolutionary cancer treatment using genomic tests. I hope all these tests will be useful, and these tests can be used judiciously and appropriately based on the sample availability, based on the financial capability of the patients. So ALPS is a one-stop center where the MyGenome is located. Where we do the genome sequencing. It's ISO 13485 accredited facility. At ALPS, we also have a CGMP facility for mesenchymal stem cells, and we have a full-fledged medical center, and we also have a wellness center, and which is located in the heart of the Kuala Lumpur near KLCC. So for collaboration or partnerships and the underlying numbers, people can, my email ID is also there in the first slide. So if anyone is interested, can contact our marketing team. Hi, Jason.
Very interesting topic. Demystifying cancer. So for Q&A, it's for okay. Let me try to fix it. All right, is it better now?
Yeah.
All right, great. So, yeah, is there any questions from the floor? Okay, there is one question that came in from our Facebook page, actually, Prof. Manikan Ravichandran. So one of the questions they asked, one of the doctors asked is a district medical officer working in Kuala Lumpur is actually asking, what is the best type of investigation, genome investigation for cancer?
Of course, the best first line of investigation is the histopathology, and in terms of genomic tests, now you have plenty of FDA approved checkpoint inhibitors, and you have a lot of other things are available. So so they have to decide based on the need of the patient. Actually, best type of test depends on the affordability and the need of the patient. Sometimes the samples are very difficult to obtain, so depends on that they have to choose. So before they choose, they need to know whether they want to, if they know the tumor type, I think they can focus on the certain panel of genes they can look for. Or if they do not know, they can go for the whole genome sequencing, and then they get the full overview of the whole disease in the patient.
Right. Thank you, Prof. Manikan Ravichandran. And thank you so much for the question, Dr. Ravi from Kuala Lumpur. All right. I think there's one more question, Prof. Manikan Ravichandran. Are you okay to answer that? So this next question is this next question is actually from Dr. Eileen. She's from Hospital Denom in Sabah. She's asking, Prof. Manikan Ravichandran, if I was to do this test and if I had some genetic predisposition or some risk factor for cancer, how will that affect my insurance? Is there any way that your organization is helping with that?
Genetic predisposition. Okay, when you do the whole genome sequencing, you will know the whole genome, you know, your risk for all the disease, not only cancer, you get everything out. I'm not sure about this insurance industry, whether they are looking at your genome or not. If they look at your genome, they will increase the risk. Definitely the premium might change. But I'm not an insurance agency, but definitely your genome test will reveal you and tell you clearly a risk. But don't get scared of it. You will, depends on your lifestyle change and epigenetic change, it might not even happen. So, I'm not here to answer about the insurance industry.
All right. Great. So Prof. Manikam, fantastic. There's actually two more questions from the floor. Thank you so much for asking. This question is actually from Dr. Murali. He's actually asking, Prof. Ravi, what is NGS, the next genomics sequencing? Can you just explain a little bit more about that?
NGS is a next generation sequencing. It's a technology, actually. It is a sequencing technology which you can use to sequence the whole genome. And it's same like WGS, where you can sequence the whole genome and you get the whole all the 30 billion bases can be sequenced. Okay. I think whenever you want to go for a test, you ask for the coverage. How many times the genome is covered? Let's say you're going for a whole genome sequencing. At MyGenome, we have 100 times coverage. Some of them will offer very cheap sequencing, but they will do it in a very low coverage, maybe 10 times coverage or 3 times coverage and so on. So it's very, very difficult. We are in the actually the threshold of this revolutionary era. Everyone is learning. Everyone is learning, and too many things are coming in. So only thing I can advise is just ask for the accuracy and the coverage, and you balance with the cost of available fund, and you can choose the test based on that.
Thank you so much, Dr. Murali, for the answer, and thank you, Prof. Manika Chandra, for that answer. Actually, Dr. Chu asked a similar question regarding insurance, but you've already answered that, so I don't think we can go for that question. But there's one more question from a Dr. Sharo. He's actually asking, what's the role of genomics and epigenetics in precision medicine?
Okay. We know genomics causes mutation. At the genomics test, we know if there is any changes happens in the cell, and by genomic test, we can predict are we prone to cancer. It depends on the cell. If you're going to look at in the blood, it is the germline mutation. So it tells you the possibility, the probability of getting cancer or any other diseases. That is genomic. Epigenetics is basically methylation changes, some minor changes on the DNA that also tell you your possibility of probability of getting cancer or other non-communicable diseases and so on. So both genomics and epigenetics test definitely help in predicting our future health.
Thank you so much, Prof. Manika, for that question. I hope Dr. Sharo that answered your question. If anything else, you can always contact Prof. Manika Ravichandran personally, and you can ask him more details from there. This is the same for the rest of the watchers at home. Please contact Prof. Manika Ravichandran or else directly if you have any questions or you want any partnerships. Well, unfortunately, there doesn't seem to be any more questions coming in. So I think it's good time and it's an apt time for us to end this session. Thank you so much, Prof. Manika Ravichandran, for the exciting talk earlier about demystifying cancer. Again, much appreciated for your talk here and your input on how we can move forward in terms of cancer research and cancer treatment. Once again, thank you so much, Prof. Manika Ravichandran, and thank you to all our viewers who are watching today. All right, have a good day, everyone. Bye.
Have a great weekend.